what are the catalysts? Catalysts are substances because of their presence, rate of chemical reaction changes without undergoing any chemical change to it. That means the substance what we are adding to that substance, no change will be there. Or we can say the substance what we are adding, that is known as a catalyst. There will be other reactants, along with that we are adding a substance. That substance doesn't get any change. But because of the addition of this catalyst, reaction will be quick, or quick reaction will take place. There are number of chemical catalysts. In addition to that, nowadays biological catalysts are also using by chemical industries. Now few examples of chemical catalysts I'll say. One is manganese dioxide, other one potassium permanganate, then potassium dichromate K2Cr2O7. These all are chemical catalysts. Now, biological catalysts are known as microbial enzymes. Now, microbial enzymes, for that some examples we are having, these all are acting like a catalyst. They are oxidoreductases, transferases, hydrolases, lyases, isomerases, ligases, etc. Number of, many are there. Only few examples we need to study. They are oxidoreductases, transferases, hydrolases, lyases, isomerases, then ligases, etc. These are the examples of microbial enzymes. They are acting like catalysts. Now, by using microbial enzymes as catalyst, number of industries are working. Those industries name I am saying leather industry, because of the help of microbial enzymes we can say that many use is there to it. Microbial enzymes are using for various or in various industries. They are leather industry, cheese industry, plant extracts industry. That means some juice, syrup we people are removing with the help of microbial enzymes. Then textile industry, that is cloth industry, paper industry, etc. Like that way in various industries microbial enzymes are using. Microbial enzymes are also catalysts. They are known as biological catalysts. Now, when we use such a catalyst, we can say biological catalyst, or we can say as microbial enzymes. Some advantage is occurring in the reaction. So we can say that what are the advantages in the use of microbial enzymes? First use, they are active. They will function even at a low temperature, low pH value, low pressure. Less pressure will be there, less pH value will be there, less temperature will be there, even then they can function. Second thing, by using microbial enzymes, we can save energy. Why? Because heating process is not required. Because of that we can save energy. Other thing, there is no necessity of using erosion proof instruments. That means the glass apparatus using necessity is not there. Any instrument, any vessel we can be able to use it. Then we are not heating it. Low temperature it can function, low pressure it can function, even low pH value it can function. So because of that we can say that even there is no production of any pollutants, we can say. No pollutants production will be there because of the addition of which one? Microbial enzymes. As a result of it we can say that the use of microbial enzymes are eco-friendly. It is not going to harm our environment. Now, one more thing is there, that is because of the addition of enzymes, the detergents, we can say that we are familiar with the detergents. Detergents are using for washing our clothes. In that detergents, they are adding enzymes. With the help of these enzymes, dirt or muck we can say, these things easily removed from the surface. Second thing we can say that glucose as well as fructose, which are producing from corn flour, is happening because of the help of enzymes which are producing by bacilli as well as streptomyces bacteria. Production of glucose as well as fructose from the corn flour is happening because of the enzymes producing by the bacilli as well as streptomyces bacteria. Various organic acids, or we can say amino acids, are used for the production of various products. Now we can say that here for the production of various organic acids, various microbes are also used. Microbes, it's a meaning microorganisms. So here now we are going to study various organic acids. These organic acids we can call in other name, that is amino acids, to them producing by different microorganisms. The organic acids which are producing by microbes or microorganisms are used in various industries we can say, or for the production of various things. Let us see now. Here these are the different types of microbes. Here Brevibacterium as well as Corynebacterium. These two are different types of bacterium. First one Brevibacterium, second one Corynebacterium. Here this is Aspergillus niger. This is a type of fungus, it is fungus. Aspergillus niger. Here Lactobacillus delbrueckii. This is a type of bacterium. Lactobacillus delbrueckii. It is a type of bacterium. Now we can say that a type of bacteria. This is Aspergillus itaconius. This is a type of fungus. So here now Brevibacterium, Corynebacterium, Aspergillus niger, Lactobacillus delbrueckii, Aspergillus itaconius. These all are the microbes, microorganisms. Maybe fungi, or we can say maybe bacteria. Now here which all acids, or we can say which all amino acids they are making. First one Brevibacterium as well as Corynebacterium. Both of them are producing L-glutamic acid. L-glutamic acid. Aspergillus niger producing two types of acids. One is citric acid, other one is known as gluconic acid. So here Aspergillus niger producing two types of acids, two types of amino acids. They are citric acid as well as gluconic acid. Then Lactobacillus delbrueckii producing lactic acid. Aspergillus itaconius producing itaconic acid. All these are amino acids. Now all these acids are using for the production of different substances. These all substances are having use in the production of, or we can say these all acids are using for the production of various commercial products, we can say. Here glutamic acid, L-glutamic acid is using for the production of Ajinomoto. Its other name is monosodium glutamate. Second one, citric acid is utilizing for the production of various drinks. Then we can say toffee making and chocolate making work. Then gluconic acid is utilizing for the production of minerals. We can say that used as a supplement for calcium as well as iron. Calcium as well as iron minerals production, this gluconic acid is using. Lactic acid, it is using for the production of vitamins. Even we can say lactic acid is a source of nitrogen. In the lactic acid, nitrogen will be there. Even lactic acid is utilizing for the production of vitamins. Now itaconic acid is utilizing in paper, textile as well as plastic industry. Even it is using for the production of gum. So here we will see here different types of microorganisms given, which all types of amino acid or organic acid they are producing given, for the production of which substances they are helping, that is also given. First we will see Brevibacterium as well as Corynebacterium utilizing for the production of a glutamic, L-glutamic acid. It is utilizing for the production of Ajinomoto or monosodium glutamate. Aspergillus niger is utilizing for the production of citric acid as well as gluconic acid. Citric acid is utilizing for the production of various drinks, then toffees as well as chocolate making it is using. Gluconic acid is using for the production of minerals, like we can say calcium as well as iron. Other one, now Lactobacillus delbrueckii is utilizing for the production of lactic acid, and this lactic acid is a source of nitrogen as well as it is utilizing for the production of vitamins. Aspergillus itaconius is utilizing for the production of itaconic acid. It is utilizing or using in paper, textile and plastic industry as well as it is utilizing for the production of gum. What are the various sources of these microbes? That means where all these microbes are growing. Here now Brevibacterium, Corynebacterium, these two types of bacterium are growing in sugar molasses as well as sugar beet molasses, then ammonia salt. Sugar molasses, that means in sugar refinery, after separating the sugar or after crystallizing the sugar or after refining the sugar, the waste product, very thick substance will be there. A waste of the sugar we can say. It will be a very dark liquid, very viscous, very thick liquid will be there. That is known as molasses. Here now sugar beet molasses. Sugar beet means beetroot plant, all are knowing. Beetroot, that means we can say that table beet or the beetroot curry we people are making. So that beetroot same genus we can say or same name only to the sugar beet plant. But at the same time this sugar beet plant that is having large quantity of sugar in it. So where sugar industry is there even they are growing. Where sugar cane growth is taking place, where people are growing sugar cane plants, there even people are growing sweet or what we can say sugar beet plants. They are almost looking like a beetroot plant, beetroot, but color variation is there. Beetroot, its color is dark pinkish. But here this sugar beet plant, sugar beet, its color is more or less whitish. From that sugar producing large quantities. So we can say that where the work of sugar production is happening. Normally we know sugar is producing from sugarcane plant. So here we can say that not only from sugarcane plant but also from sugar beet plant. When they remove sugar from sugar beet plant, again there will be the waste product of very dark thick liquid that is known as beet molasses. In these two molasses, Brevibacterium as well as Corynebacterium grows. Even we can say salts of ammonia. In that also this Brevibacterium as well as Corynebacterium grows. Now these bacteria are helpful for the production of amino acid L-glutamic acid. Now next one, Aspergillus niger. That is a type of fungus. It is helping for, means it is growing in two types of medium I can say. One is sugar molasses as well as salt medium, or we can say glucose then corn steep liquor medium. This either this medium or either this medium we can say. So we can say that or either in this or in this. Any of these Aspergillus niger can grow. So Aspergillus niger is a type of fungus. It can grow either in sugar molasses as well as salt. In the salt also it can grow. Even we can say that glucose as well as corn steep liquor. Now what is corn steep liquor I will say you. Before that I am saying in the salt or in the sugar molasses growing Aspergillus niger is helpful for the production of citric acid. The Aspergillus niger which is growing in the glucose as well as we can say corn steep liquor, either corn steep liquor or in glucose growing Aspergillus niger is useful for the production of a type of amino acid that is known as gluconic acid. Now we will study what is corn steep liquor. There is corn industry I can say. Corn mill is there, corn industry is there. In the corn industry various products are making. In the corn industry many products are producing, for example gluten meal, starches, proteins, alcohol, even sweeteners. Such type of substances are making in corn industry. When they make these all substances, when in such industry these all things making, that time some waste products also producing. The waste products are bran. Bran means the outer cover of the corn. Germ. Germ means the inside, we can say carbohydrate substance is there. Inside central region of the corn which is there, that is known as germ. Then one more thing, one more waste is producing, that is corn steep liquor, a waste product. After producing all these things, there a very dark thick liquid is forming. That is the waste product we can say. That waste product is known as corn steep liquor. Here you people studied sugar molasses, then sugar beet molasses, then we can say corn steep liquor. All these things are very dark thick liquid it is. Now here we studied that sugar molasses, then sugar beet molasses, ammonium salt. In that Brevibacterium as well as Corynebacterium are growing. Now here we can say that in corn steep liquor as well as glucose, here we again we are seeing corn steep liquor. Again here we are seeing corn steep liquor. In them different types of microbes are growing. One is Aspergillus niger, Lactobacillus delbrueckii, Aspergillus itaconius. All these microbes or microorganisms are growing corn steep liquor, corn steep liquor, corn steep liquor. In this medium these three types of microbes can grow. Aspergillus niger can grow either constip liquor or glucose. Or we studied here sugar molasses as well as salt. In that also it can grow. Now Lactobacillus delbrueckii can grow in molasses, any molasses, sugar molasses or beet molasses. Then constip liquor. Then Aspergillus itaconius can grow molasses mentioned, that is either sugar molasses or sugar beet molasses, then constip liquor. It is a byproduct or the waste matter, we can say, which is obtaining in the sugar, or we can say that in the corn industry, we can say. So there a waste product like a constip liquor will get. So in that Aspergillus itaconius, Lactobacillus delbrueckii, Aspergillus niger, all these microbes can grow. Now Aspergillus niger, which is growing in sugar as well as sugar molasses as well as salt, that is helping for the production of citric acid. Aspergillus niger, which is growing in the glucose as well as constip liquor, it is helping for the production of gluconic acid. Lactobacillus delbrueckii, which is growing in molasses as well as corn steep liquor, helping for the production of lactic acid. Aspergillus itaconius, growing in molasses as well as corn steep liquor, helping for the production of itaconic acid. So here we can say that as per our wish, we can be able to grow all these microbes in certain medium, certain solution, we can say. In that they will grow as per our requirement. We can utilize them for the production of various organic acids. These all organic acids are helpful for the production of various products. We can say various commercial products. What is xanthan gum? Xanthan gum is a gum. It is made with the help of a particular species of bacteria, that is known as Xanthomonas bacteria. Xanthomonas bacteria. With the help of this Xanthomonas bacteria, xanthan gum is producing. Xanthan gum. Now, this xanthan gum is giving or imparts thickness to various food items like ice creams, puddings, chocolates, milkshakes, chocolate drinks, instant soups, etc. To various substances, food items, we can say various food items, this xanthan gum gives thickness. Now, which all food items it gives thickness? Ice creams, puddings, chocolates, chocolate drinks, milkshakes, instant soups, etc. Now, this xanthan gum is useful for the production of different types of pigments. We can say different types of colors. Then for the production of fertilizers, weedicides. Weedicides means like a pesticide. Pesticide for killing pests. We can say mosquitoes, cockroaches, for killing them. Pesticides we are using. Weedicide means in the crop field along with crops. Crops are growing in the field. Along with the crops, grasses will be there. Those grasses harms the crop plants. So for killing the grasses, we are spraying weedicides. Then xanthan gum is used for the production of textile pigments. Textile means a cloth. For on clothes, different color want to put it. For that pigments are making, or colors requirement is there. Such colors, different colors we people are putting on the clothes. So for making such colors, xanthan gum is using. Then we can say that for the production of different types of toothpaste, xanthan gum is using. Then we can say that for the production of high quality paper, we are using xanthan gum. Now, for the production of xanthan gum, we are using a particular type of bacteria. That bacteria is known as Xanthomonas bacteria. Now, we can grow Xanthomonas bacteria in a medium. That medium, we can say that starch medium as well as molasses medium. In that, this Xanthomonas species can grow and they can be able to make xanthan gum. That xanthan gum is adding in various types of foodstuffs as well as for making various types of products we are using it. Here different substances are shown. These all substances are produced because of the action of microorganisms, we can say. So these all things are produced by the processing of microbes. Here now we can see various substances. All these substances are produced by microorganisms or microbes. Because of them, so many substances are made. Now, with the help of all these substances, different purpose we can be able to use them. All these substances we can use them for different functions or different purposes. So we can say these all substances have different roles. So these all substances are made by microbes. So we can say that substances processed by microbes. Then all these substances are helping for different purposes. So what are the roles of all these substances? That is given here. Here we can say that because of microbes, different substances processed or microbial processing we can say. As a result of it, various substances are produced. Now let us see. Here citric acid, malic acid as well as lactic acid. These are produced by microorganisms. Then here glutamic acid, lysine as well as tryptophan. This is also produced by microorganisms. Then here niacin and natamycin. Other chemicals produced by microorganisms. Ascorbic acid. This is a type of vitamin. Vitamin C we can say. Vitamin 2, vitamin 12, B12. That is also produced by microorganisms. Then beta carotene, lycopene, xanthine as well as lutein produced by microorganisms. Then here polysaccharides, glycolipids produced by microorganisms. Here vanillin, ethyl butyrate, that is a type of fruit flavor. Then peppermint flavor, then essence of various fruits and flavors. All these things are made because of the processing of microbes. Then here xylitol as well as aspartame, TAME, aspartame. This is also produced by microorganisms. Now, what are the different roles of these all substances made by microorganisms or because of the processing of microorganisms? We'll see. Citric acid, malic acid as well as lactic acid, that helps, they give acidity to the food item. So here we can say that impart means they give acidity to the food items. Then here glutamic, lysine as well as tryptophan helps for protein binding inside the cell or we can say inside the protein. So it is helping for binding, attaching proteinaceous substances inside the blood or inside the cell. Niacin as well as natamycin helping for microbial restrictor. That means that they prevent the overgrowth of microorganisms. They protect us from over or more production or they control the overproduction of microorganisms. Ascorbic acid, that is vitamin C, vitamin B2 and vitamin B12 are acting like antioxidants. That means they won't help the oxygens to react with the food items. As well as we can say that they help for the production of vitamins. We can say that these all are the vitamins, so they help for the production of the vitamins. So because of the help of microbes, such vitamins are producing. So vitamins production helping, even because of the help of these vitamins, there won't have a reaction with the oxygen. That means if oxygen react with the food items, food item may get spoiled. So we can say that they are acting like antioxidants. Because of that, our food material will be getting protected. Then beta carotene, lycopenes, xanthines, luteins, they are helping for, or these products produced by the microbes, microbial processing. As a result of it, we are getting edible colors. Now polysaccharides, glycolipids, they are the additives, or we can say preservatives, or we can say they are the emulsifiers. So with the help of that, our food material will be getting protected. Now vanillin, then ethyl butyrate, that is fruit flavor, peppermint flavor, essence of various fruits and flowers. These all are essence. These essence also making because of the microbial processing of different microbes. Then xylitol, aspartame, these substances helping for making artificial sweeteners. These substances are made by the microbes. With the help of that, we are getting artificial sweeteners. It's a specialty sugar. Our sugar, what we are using at home, that is having high calorie, but at the same time artificial sweeteners has less calorie. Because of that, it is not going to harm us. So the people, those who are having diabetes, they can use artificial sweeteners in their tea, coffee, etc. As a result of it, they will feel sweetness. At the same time, it is having very low calorie. Because of that, it won't affect their health. What are antibiotics? Antibiotics are medicines which are produced from bacteria as well as fungus. Here we have different types of antibiotics. These antibiotics help to cure, or we can say help to control, or we can say help to destroy gram positive and gram negative bacteria. Gram positive and gram negative bacteria. Now, what are antibiotics? Antibiotics are medicines which cure or which destroy the disease germs which are causing diseases to human beings as well as animals. These antibiotics are making from different types of bacteria as well as fungus. Here now we are having number of antibiotics which can destroy gram positive and gram negative bacteria. Now here penicillin, cephalosporins, monobactam, bacitracin, erythromycin, tetracyclines, gentamicin, neomycin, streptomycin, vancomycin. These are the different types of antibiotics which are helping for destroying various strains or different strains of gram positive as well as gram negative bacteria. Now here we are having another antibiotic that is known as rifamycin. This is an effective medicine for tuberculosis. With the help of rifamycin, people, those who are suffering from tuberculosis, getting cure. Now, what is meant by antibiotics? Antibiotics are medicines which are producing from bacteria as well as fungus, which help to control the growth of microorganisms. As a result of the growth of microorganisms, various diseases are getting to human beings as well as animals. For controlling the growth or for destroying the microorganisms which are growing in the human beings or animals, we are using antibiotics. Now, for getting or for causing different diseases, gram positive as well as gram negative microorganisms are helping. So for controlling their growth, these all antibiotics are helping. We can say that in human beings alone, not even in animals, various diseases are causing by these bacteria or different types of microorganisms. Now these bacteria, that is gram positive bacteria as well as gram negative bacteria, we can say that gram meant for a kind of test it is. So here this is the test of gram positive test, gram negative test. Some bacteria can be detected with the help of gram positive test. Some bacteria can be detected with the help of gram negative test. Now these gram positive as well as gram negative bacteria can be cured with the help of these antibiotics. Once again we will study about the names of antibiotics which can cure or which can control or which can destroy this gram positive as well as gram negative bacteria. Penicillin, cephalosporins, monobactam, bacitracin, erythromycin, tetracyclines, gentamicin, neomycin, streptomycin as well as vancomycin. Now here rifamycin, other type of antibiotic which is having effective or which is very much effective against tuberculosis. So the people, those who are having tuberculosis, to them this kind of antibiotics are given by the doctors. As a result of it, they are getting cure from tuberculosis. Microbes and fuels. With the help of microbes, different types of fuels can be obtained. First one we can say as gaseous fuels. Gaseous fuels are produced by the microbial anaerobic decomposition of urban agricultural and industrial waste materials. So here gaseous fuels are produced by microbes by anaerobic respiration. As a result of it, they decompose urban agricultural waste products as well as industrial waste products. As a result of it, a gas is coming out. That gas is known as methane gas. Methane gas is a fuel. So we can say that as a result of microbial decomposition of urban agricultural waste as well as industrial waste, a gas is producing by microorganisms. That gas is known as methane gas. It is a fuel. So we can say that methane gas is a gaseous fuel that is obtained because of the anaerobic process or anaerobic respiration done by the microorganisms. As a result of it, they decompose urban agricultural waste materials, as well as industrial waste materials. As a result of it, they produce a gas. That gas is known as methane gas. This methane gas is a fuel. This methane gas is a gas, so we can say that it is an example of gaseous fuel produced by microorganisms. Second thing, ethanol. Ethanol is a fuel. We can say that it is a clean fuel. Again, we can say that smokeless fuel. Smokeless. Even we can say clean fuel. Again, we can say that it is a type of alcohol. So we can say that ethanol is an alcohol and it is a clean fuel as well as it is a smokeless fuel that is produced by the yeast. Its name is, this is produced by the help of a particular type of yeast. So ethanol is a type of alcohol that is produced by a yeast. That yeast is known as Saccharomyces. Saccharomyces. With the help of yeast Saccharomyces, ethanol, a type of alcohol, is produced. It is a smokeless as well as clean fuel. Now other one, that is hydrogen gas. Hydrogen gas is considered as the fuel of future. In future, hydrogen gas will be the fuel. Then, how it is produced? Because of the biophotolysis of water. Biophotolysis of water. This biophotolysis of water is doing by bacteria. By photoreduction. Photoreduction. So here hydrogen gas is considered as the future fuel that can be obtained with the help of bacteria by biophotolysis of water. With the help of this process, we can obtain hydrogen gas in future. For that, bacteria is helping. Now, which work bacteria is doing? Bacteria is doing photoreduction process. As a result of it, water split into hydrogen as well as oxygen. That hydrogen can be utilized for the fuel. Oxygen liberates into the or escapes into the atmosphere. Then that we can utilize for breathing. What is meant by renewable source of energy? Renewable source of energy means the source of energy which is available again and again. It is not having any end. Here we are going to study about biofuel. Biofuel is an important among renewable source of energy. Here we can say that even sun is a renewable source of energy. Similarly, biofuel also renewable source of energy. Then here there are different types of biofuels. They are solid, liquid as well as gaseous. Solid type of biofuels are coal, dung, then crop residue. Crop residue means rice plant, wheat plant. After harvesting, the waste product occurring, that is known as a crop residue. Then here liquid fuel, that is vegetable oils as well as alcohol, ethanol, all we can say. Then gaseous fuel, that is gobar gas. Gobar gas got other name, that is biogas and coal gas. So here different types of biofuels are given. In different forms it is available, solid form, liquid form as well as gaseous form. Now what are the advantages of using of biofuels? One advantage of using biofuel, we can say that it is eco-friendly. Second thing, we can say that available abundantly or plenty. Biofuel is available abundantly and production is very easy. We can say that we can produce easily. Easy production we can write. So these are the advantages of using of biofuels. What are the advantages of using of biofuels? First thing, they are eco-friendly, it is not going to harm our environment. Second thing, it is available plenty, large quantities of biofuels are available. Third thing, production of a biofuel is very easy. Solid waste, liquid waste as well as other type of waste are increasing day by day. As a result of it, there occurs increase in the pollution. If increase in the pollution, there will be increase in the diseases. For controlling pollution, microbes play a big role. You all are aware of biogas plant as well as compost production. Biogas plant as well as compost production. In the biogas plant, microbes are acting or working. They help for decomposing all waste products. Even in compost production, compost pit, there we are adding waste products. It's a decomposition also done with the help of microorganisms. Now, in cities or we can say urban areas, there so much sewage as well as waste products are collecting. We will study now how we can be able to dispose them. Here we are going to study the disposal of urban waste with the help of microbes, how it is possible. How to make landfill or landfilling sites. For making landfilling sites, we have to follow different steps. Now we can say that first we have to select a place, an open place where residential area is not there. Such a large as well as open space we have to select. There people should not stay. Such area we have to select it. There we have to dig and make a large pit. Then inside the pit as well as bottom part of the pit, we have to stick a large plastic sheet. And then inside that sheet we have to fill compact condition, that means pressed condition, large quantity. Why we people are pressing it? Because large quantities of waste matter which is collecting by the garbage vans, that thing can be able to dump it inside such a large pit. Then pressing process should be done, otherwise means we cannot be able to fill large quantities of waste inside. Now here one figure shown. This is an example of modern landfill site. So here in this a methane gas collecting one pipe system is shown. Then the waste products we have to fill it inside. Then that its processing process will be done. Afterwards we have to collect the garbage. Changes to after few days the garbage turns to compost. That compost they are collecting it and they can be able to utilize for putting to the plants. Once the garbage collection garbage changes to compost, that thing they can be collect it and put it to the plants. Other thing here one pipe system is there, through that biogas or methane gas is coming out. That gas they can be able to collect and use it as a fuel. So this is the modern landfill site. This is making where people are not staying, such a open place. They are digging a large well like a pit they are making. Then in that they are dumping all the waste products. We can say that means they are all garbage, that is degradable type. Degradable garbage we have to put it. Degradable, it's a meaning the waste products which can be decomposed by microorganisms. Such a type of waste matter we have to put it inside this well. Then before putting the waste products, it should be lined with a plastic sheet. Why to lined with the plastic sheet? Because in the waste matter so much toxic substances can be there. Many toxic substances will be there. If we are not lining the pit with the plastic sheet, there can be the leaching of toxic substances into the soil. What is meant by leaching? Leaching is the leaking, we can say, or going the waste products going deep inside all the toxic substances which are present inside the waste. Then for protecting the soil, we are covering or lining this pit, the well, we can say, that well with the plastic sheet. Then the reactions, everything will take place inside this large pit. Microorganisms will react with the waste products. They decompose it. From that a gaseous matter will come out. That gas will be collecting through this, going out through this pipe, and it will be collecting in a tank or in a cylinder, we can say. Then the waste products, once the gas removal will occur, then the slurry, the waste product what is forming, or the compost what is occurring here, that is collecting through this pipe and its processing will be done. That means it can be able to take it and put it to the plants. Then once its use will over, once the production of a compost will be done, gas removal work will be done, we can remove out all these things. Then again we can fill it with the waste products or sewage, we can say. Then again this process occur continuously. As a result of it, the gas, the biogas which is required for the production of electricity, or we can say the gas cylinders. You can find that in Mumbai different areas gas is coming. That means a natural gas supply is taking place through pipeline system, we can say. That gas is passing by making such a type of a biogas plant. So the gas which is getting, so we have no need of buying gas cylinders. Directly through pipe system gas is coming to their houses. So for that all means such a landfill site can help. Gas also can collect, even the compost production also possible. Then here inside middle place, Such a large pit will be there. In the middle some bioreactors they are keeping it. As a result of bioreactor means bioreactors are the apparatus. With the help of bioreactor we can say that some harmful microorganisms which are present inside, we can say cholera germs, then typhoid germs, which are present inside this pit can be killed or can be destroyed. Then now how the filling process is taking place, that we will study step by step here. Now first what we are doing, first we are making a very large pit. After that the pit is lined with plastic sheets. Then we are dumping degradable waste inside. Now the degradable waste is covered with layers of soil, sawdust, leafy waste, even we can say some biochemicals also they are putting and covering the pit. Then microorganisms present in the soil, that means the microbes means microorganisms which are present in the soil as well as top layers that will decompose the waste products which are putting inside. Then completely filled pit is sealed with slurry. Slurry means what? Mud plus water. So here we can say that already it's a filling work done, very much compressed condition, pressing we are filling the waste products inside the pit. Once or when that pit will be filled, it will cover with the leafy leaves we can say. Then sawdust, then mud will be there. Even we can say some biochemicals also they are spreading it. After that what is there? Above that they are making slurry with mud. That means water plus a mud mixture will be there. That they are mixing it. Then it won't be so thick, it won't be so thin. Such a condition slurry they are making. Then that they are spreading over the pit. Then within few days a best quality compost will occur, as well as here now gas collecting pipe is there, then gas collection also will occur. Which gas is collecting? Methane gas is collecting. That methane gas we can utilize for burning process in our home. Means here we are not using it, but where people are getting gas through pipeline, so there such type of gas they can be able to utilize. All are knowing about the sewage. Sewage means waste products. Waste products are producing in villages as well as in cities. In villages the sewage which is producing, that they are putting on the soil or to the plants or to the trees. At the same time here in cities the sewage we have to carry it to the processing units. There microbes or microorganisms acted upon it. Now when sewage carried to the sewage carrying units or different places where we people are dumping in it, so such areas there they are adding some microbes which can be able to decompose some compounds, as well as we can say that harmful microorganisms, like we can say typhoid causing germs as well as cholera causing germs. For killing them they are adding some microbes which can be able to decompose harmful compounds as well as different types of harmful microorganisms. As a result of it they produce methane, CH4, methane as well as carbon dioxide gas. This methane gas we can collect as biogas. Then one more important thing, here phenol oxidizing bacteria also adding into the sewage. As a result of it, decomposition of xenobiotic chemicals which are there inside the sewage also will take place. So in the sewage, some poisonous chemicals will be there. For decomposing that, different types of microbes are helping. We can say that a type of microorganism which decomposes some compounds, as well as we can say poisonous or harmful disease germs of cholera as well as typhoid, to them also can be able to destroy such a type of microbes they are adding into the sewage. Other thing, phenol oxidizing bacteria, a type of microbe they are adding into the sewage. As a result of it, decomposition of xenobiotic chemicals which are present in the sewage also takes place. In the sewage processing unit, there will be solid waste matter. That waste matter is known as sludge, that we can utilize as fertilizers. Second thing, microorganisms acting or decomposing the waste products. As a result of it, there will be the formation of water. That water is not harmful. It is safe to use. Other thing, the microbes which are helping for bioremediation. We can say that breaking down of, we are using microbes for the bioremediation. Bioremediation, its meaning, breaking down of microbes which are polluting the environment. So as a result of microbes, we are protecting the environment. Or there are harmful microbes as well as useful microbes. Harmful microbes destroying the environment, or because of the help of other microbes, bioremediation process is taking place. That means breaking down of harmful microbes, or we can say destruction of harmful microbes. As a result of it, there occurs control in the pollution of our environment. Human beings made so much progress in the technology. At the same time, how much progress is made in technology, that much level of pollution also increased. Now, in the water, air as well as soil, so much pollution occurred. With the help of microorganisms, there are various ways for controlling pollution which is occurring in the air, water as well as soil. Now let us see which all microorganisms can help for reducing the pollution in different sources. Here now different types of microbes name given. Here Theobacilli and Sulfobacilli. This is the name of two types of bacteria. Here Pseudomonas spp, one type of bacteria, and Alcanovorax borkumensis. This is other type of microbe. Vibrio, Idionella succaensis. This is other type of microorganism. Here Actinomycetes, Streptomyces, Nocardia, Actinoplanes. These are different types of microbes. Acidophilium spp, one type of microorganism. Acidobacillus ferroxidens, other type of microorganism. Then Geobacter, other type of microorganism. These all microorganisms are used for reducing the pollution in different ways. Now which all actions these all microbes are doing, we'll see now. First, Theobacilli and Sulfobacilli. These microbes convert metals like copper, iron, zinc, uranium, etc. into compounds before leaching. So these microbes are having the capacity to convert metals like iron, then we can say copper, uranium as well as zinc into its compounds. Before leaching means before going deep into the soil. So here Theobacilli as well as Sulfobacilli, these type of microorganisms, they have the capacity to convert metals like copper, iron, then zinc as well as uranium into their compounds before leaching. Leaching means before going deep into the soil. Now, Pseudomonas spp as well as Alcanovorax borcumensis, these two types of microbes helping for clear oil spills in seawater due to some problems happening. We can say that ship which is carrying oils, oil tanks, sometimes some leakage may occur. As a result of it, oil layer may occur on the sea. So that oil spill, want to remove it. For that, these microbes can help. They can clear the oil spills, oil which is fallen in the seawater or river water, that can be able to absorb with the help of these microbes. They are Pseudomonas spp and Alcanovorax borcumensis. These two types of microorganisms can clear the oil spills. Vibrio Idionella succensis. Vibrio Idionella succensis. This is one microorganism. This is other microorganism. They are having the capacity to decompose PET. PET means polyethylene terephthalate. Polyethylene terephthalate polyester. Polyethylene terephthalate polyester. Its decomposition, Vibrio as well as Idionella succensis microbes are helping. Actinomycetes, Streptomyces, Nocardia, then actinoplanes. Actinomycetes, Streptomyces, Nocardia, actinoplanes. These microorganisms helping for the decomposition of rubber from garbage. In the garbage, there can be rubber particles. So there, the rubber particles decomposition, these microorganisms are helping. Actinomycetes, Streptomyces, Nocardia, actinoplanes. These microorganisms are helping for the decomposition of rubber particles which are present in the garbage. Now, Acidophilium spp and Acidobacillus ferroxidans. These microorganisms control soil pollution occurring due to acid rain. Because of the help of acid rain, soil get polluted. Then, for controlling that soil pollution, Acidophilium spp and Acidobacillus ferroxidans microbes are helping. Now, Geobacter is a microorganism which is helping for converting salts of uranium into insoluble salts. So, convert this Geobacter microbe or Geobacter bacteria we can say, which convert salts of uranium into insoluble salt. As a result of it, it prevents mixing of uranium with groundwater resources. So here what we can say, conversion of uranium into insoluble salt. As a result of it, there won't have mixing of uranium with water. For that, Geobacter bacteria is helping. What are inoculants? Inoculants are vaccines or serum we can say, or injections. Some of the microorganisms containing inoculants, that is produced as a result of fermentation process. These inoculants are sprayed on seeds or released into the plants. As a result of it, plants grow well. Even we can say that the quality of vegetarian food increases. Solution of Acetobacter bacteria as well as artificial nitrogenase helping for organic farming. Organic farming. So here solution of acetobacter and artificial nitrogenase helping for organic farming. Chemical fertilizers causes soil pollution. For preventing soil pollution, we can add solution of acetobacter and artificial nitrogenase in the soil. As a result of it, the pollution created by the addition of chemical fertilizers can be prevented. When we spray chemical pesticides into the soil, fluoroacetamide like chemicals are adding in the soil. They are harmful to the soil. For protecting the soil from such chemicals, some microbes are helping. What are bioinsecticides? Bioinsecticides are the insecticides made from microbes. Using biotechnology, bacterial as well as fungal toxins are used for destroying pests as well as pathogens. The bacterial as well as fungal toxins we can directly add into the plants using biotechnology. Some species of fungi as well as viruses are used as pesticides. Spinosad. A byproduct of fermentation is used as a biopesticide. Thank you.